Now we are talking about transgenic animal and the main objective of this.
A transgenic animal is one that carries a foreign gene that has been introduced in its genome. And the foreign gene is elaborated using DNA. There are several methods in order to obtain transgenic animals with the insertion of foreign DNA in its genetic chains. The three most important are microinjections, retrovirus, and mother cells embryonary. The microinjections consist in the inserting of at random of gene in its genome, but only the 5% of those make a transgenic animals. This method is easily and applicable, and it is useful for several species of animals. The retrovirus, the transgene is in all of the cells of transgenic animals, and this time the gene also is inserted in at random. But there is a possibility that the retrovirus reproduces in the transgenic organisms, and it can be produce a disease. And finally, the mother cell embryonary, it is used when it's important to cure a specific sequence of the part of the genome.
The main objectives are medical and commercial. Medical, they are used to investigate certain diseases in humans and in order to then treat the illness. And they can provide antibiotics and they can give us the organ. More specifically, there are the bioreactor and the xenotransplant. The bioreactor, there are animals that have certain human genes and they produce human protein with the objective of investigate certain illnesses. And the xenotransplant, with the implantation of human genes, the animals can give us the organ. One example of this is the pig, because the DNA is similar and the organ as big as our organs. And with a commercial objective, the scientific use this animal in order to investigate the mutation in certain species. In some cases, a gene is inserted in animals to stop illnesses. And finally, here there are cases of a transgenic animal.
These are some examples of the mutation that can be produced.